Hello. Oh. All right, let's take a seat and get started. So it's my pleasure to introduce Tim Vogels as our first speaker of the session, who's going to tell us that homeostasis is it.
Hey. Good afternoon, I suppose. Not yet. Good morning. Yeah, thank you very much for the invitation. This is rather loud. If you can turn this down a little bit. I'm going to talk about homeostasis a little bit. Not really talking about homeostasis, really, but you'll see that it kind of comes close. As a definition for homeostasis, I thought this might be interesting. All the vital mechanisms, however varied they may be, have only one object, that of preserving constant the conditions of life in the internal environment, which is the condition of a free and independent life, from 1929. So I'll talk about two of these ways of keeping yourself happy internally. One by way of an intercellular mechanism, by way of synaptic plasticity, meta-learned plasticity specifically, and the other one, an intracellular mechanism by way of metabolic control of neural excitability. That's 3 papers, 2 for the first one and 1 for the other one. One of them is not published. I'm going to start with a slide that I finished with in 2011. That was the title of my talk that I flashed on in 2011. Inhibitory plasticity is awesome. At the time, I thought we had experimental evidence for this. We didn't, but we later found that other people did provide experimental evidence for this learning rule that we postulated, which shows you on the x-axis the timing difference between pre- and postsynaptic spikes, and on the y-axis the subsequent change in efficacy of that synapse. And you see a Hebbian part here in the middle, and a non-Hebbian or a pre-only part further out on the flanks that brings the efficacy down when there's only presynaptic spikes. And as I said, this was reproduced by now by multiple groups, so we're quite happy. We're also, we're delighted to find this because it had really nice qualities. This rule was a homeostatic rule in a way that it provided asynchronous and irregular activity and provided what Juliana called this morning so nicely a blanket of inhibition for any kind of memory effect that you may have in a network. So it provided stability. Now, that research is still ongoing, and there's 2 posters about this today in the poster session right after the last morning session. But it's fair to say that it may be awesome, but it's not almighty. So inhibitory plasticity is quite lonely in a way if you're using it on its own, and it's not able to do all the things. And clearly, biologically, there is many other rules in play, which experimentalists have shown multiple times, right? And we've also sort of started working with this. We've started implementing excitatory and inhibitory plasticity together. We're not the only ones doing this, but since I'm about to bash these papers, I figured I'd only show my own citations. These are 2 of them. I'd encourage you warmly to especially read the second one, which is about the interaction of excitatory and inhibitory plasticity, which can explain a lot of sort of learning effects that we see experimentally. But as we were writing these papers, all of the co-authors of these papers and another few people in my lab thought that this felt somehow incremental or piecewise because we were just contributing another one of already 50,000 papers on plasticity. And so we thought maybe it's possible to do this slightly more systematically or, in other words, with computers. So in order to do that, we first parameterized the rule that we're playing with. So for example, here a spike-time-dependent excitatory plasticity rule in its original shape. And then we introduced parameters to change that rule. So for example, the time scale of pre-post interaction were 2 parameters. Then the efficacy changes that you see in response to a pre-post pairing, both for pre-post and for post-pre. We called these gamma and kappa. These were 2 more parameters. And then we introduced another 2 parameters for pre-only or post-only effects, so for rate effects in essence. So far so good, that's 6 parameters. And then you can choose any kind of combination of these 6 parameters to come up with your own randomly chosen maybe plasticity rule, such as this one. And you can multiply this at infinitum, or just 4 times, if you will, and call those 4 rules then excitatory, 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 inhibitory, inhibitory, excitatory, and inhibitory, inhibitory. And we did just that. And then we were hoping to find, by way of genetic algorithms or some other clever search method, some sort of solution that produced stable network dynamics. And it did kind of work, and we did publish a paper about it in 2020 in line with other people's work as well that I'm mentioning down here. So far so good, only we got a polite email from the Make lab that read like this: I have read your 2020 paper and with great interest. It's the more unfortunate that your approach is bound to fail at scale. Bummer. Ta Purnima. So this is my memory of it. Purnima argues that she wasn't quite this impolite, but it was the gist of it. We sort of knew this already because we had looked at the landscape of the loss function, and we knew that it was quite tricky to navigate, and we also found that the rule solutions that we saw were quite degenerate. So there are quite a few solutions out there that were doing the same thing. And so this prompted Purnima and sorry, I'm 2 slides ahead of myself. This is what really happened. And this is why we expected it, because we had these papers that were saying exactly that it's complicated. And Purnima and Basile then ended up publishing a paper this year that basically says quite that, it's complicated. But they also started working quite intensely with each other. For 2 years, they basically finished each other's lines of code, stemming from this little note here that said, I could probably fix it for you. And they applied a method that's called simulation-based inference to scan the parameter space more thoroughly, which is to throw the dice randomly, run parameter combinations, try them out, and then estimate the entire landscape of success, or the lack of thereof, to get a better feeling of what the solution space for a set of plasticity rules looks like. And this, unfortunately, didn't work immediately because when you put all of your constraints into SBI and you say, I want networks that are asynchronous and irregular and low firing rates at the population level, low firing rates at the individual level, slow changing weights, and so on, you don't find any solutions. In fact, that's what we already knew, and Basile, my graduate student, was very close to quitting his PhD. In fact, he already announced that he was going to finish his PhD and move away. And then, as I said, Purnima and Basile started talking to each other and realized that they could just do SBI serially, really a small change that brings you from first asking for simply low firing rates globally with maybe some weird dynamics locally, and then use that posterior that you get from your inference run as a prior for the next run. So you refine your parameter space again and again and again and again, and you end up with a parameter space that actually provides the plausible dynamics that you're looking for. So that was quite neat. Mathematically not completely kosher, I was told, because we can't exclude the fact that we're missing some solutions, but we're really not interested in all the solutions, we're just interested in one, really, right? And so if you didn't like the simulation so far, the animation so far, this is really what we did. We started out with a constraint that said just low population rates, then low neuron rates, then irregular activity, slow weights, and finally plausible dynamics. And so we end up with this sweet spot of plasticity rules that give us plausible dynamics. And that it did, this search mechanism. In fact, we found quite a few plausible rules. So in total, we're up to on the order of 10,000 rules now, 10,000 rule quadruplets, that is. So it's a little bit overwhelming. You know, you used to publish a paper with just one rule at a time. So we're looking at 40,000 pages of supplementary materials. But I hope you guys can hack it when you get the paper. This flood of rules that I'm presenting you with kind of makes it seem as if this was an easy task, but I would just like to reiterate that it wasn't. In fact, the first 10,000 simulations that we didn't find any rule at all. So it was really this combined power of the different flavors of SBI that brought us there. And if you are interested in doing that kind of thing, I would warmly recommend that you talk to Basile or you talk to Jacob Mackes' lab, Pedro Gonçalves, who's now a PI in Belgium and who's helped us greatly with this and who's really the master of SBI. I'd also like to refer you to a poster that's right after this poster session that is picking up on this idea and reproducing some of our results. So maybe you're interested in that too. So now that we find 40,000 plasticity rules to play with, what are we going to do with it? I assume we're going to first look at it. We're going to do some sort of structural analysis of the rule space. And disappointingly, we found that with PCA, you see nothing. So there's no structure immediately apparent, but in fact there is structure. If you look a little bit more closely, you find that we're looking at a very complicated and tricky manifold of solutions that is well behaved, and it seems as if you can reach any one solution by foot from one other solution. We're not quite sure. We're still working on this. As you can imagine, 24 dimensional spaces are not easy to traverse, and I'm actually no topologist, so you'll have to hang on to wait for the real definite answer to this, but we're hoping this is true. Then next, we did some knockout experiments. So we just knocked out a single plasticity rule or a triplet of plasticity rules from any given quadruplet. So in this case, I'm showing you triple knockouts. We're running only one plasticity rule at a time. The other three we're setting to static. And we find that for excitatory, inhibitory, excitatory, or inhibitory rules, that means certain doom. So these rules immediately stop producing stable dynamics. Only the excitatory rules, or excitatory rules, are at all able to function in isolation. But here, a fun fact is that for those of you who hate classical SCDP, if you go on Blue Sky any day, you can find someone hating on SCDP. The SCDP solution, the classical butterfly solutions, are actually the ones that are not stable in this bar graph. If we knock out only one rule, the picture still looks surprisingly similar, especially for the inhibitory rules. It really seems as if you knock just a single inhibitory plasticity rule out, your networks immediately become much more difficult to handle in terms of simulating them. So what's the message here? Number one is coactivity of synaptic plasticity alleviates fine-tuning for the networks, but it also raises questions if studying a single rule in isolation in any given model, or similarly experimentally, though I understand experimentalists are much more bound by reality than we are, may not be super meaningful because it seems like there's a large interplay. Okay, next we looked at whether these rules that we simply picked for their homeostatic value could do anything else. So whether they were able to learn anything at all, and we started with a very simple memory experiment where we fed a single stimulus into the network. So we chose a set of neurons as an n-gram, and we played a couple of seconds, 10 seconds of a high frequency stimulus into those neurons, and then let the network rest and asked after a time whether they had a different network response to the stimulus we had already shown to the network than to a different novel stimulus. So the familiar stimulus is always the purple stimulus, and the novel stimulus is always the green one. Here's the result for a single rule. I'm showing you on the y-axis the firing rate of the entire network for this time during this testing time after a particular wait time, which is indicated here on the x-axis. And so you see that when you stimulate with a novel stimulus, you actually get a slightly higher firing rate response than with a familiar stimulus for the first 5 minutes after the original training period, and then the familiar stimulus starts showing much higher responses. So in order to quantify that for all of the rules, we actually created a measure that we call the preference, which is positive for novel stimuli preference and purple for familiar stimuli preference. And so we can plot all of the rules basically from the bird's eye view, and you see which rules or how many rules really remember anything at all, how long they remember. So the black line here is the lifetime of the memory until the stimulated response is no different from the background anymore. And you see by the color scheme whether these rules end up preferring or responding more strongly to a familiar or to a novel stimulus. What's maybe interesting to see is that virtually every neuron, every rule quadruplet that we found is really able to remember something for some time. So there's definitely a transient and sometimes up to 4 hours long imprinting effect that was not fished for during the search. Okay? Also interesting for those of you who like experimental validation, we discovered that there's quite a few papers out there actually who report similar results, but similar results in that we have so many different rules that we're quite sure to find the rule that fits your shoe. In any case, Subkin Lim and Nicolas Brunel have published something that we can reproduce, and Andrew Peters and Carandini also did. So that being said, we ran a bunch of other tasks. So we ran a transient dynamics or transient memories task. We ran a sequence learning task, a contextual novelty task. The one that I would like to focus on for the rest of the time is really the sequence learning task, because I thought the results were just quite pretty. So we did basically the same thing as with the novelty task or the familiarity task, only that we chose 6 different stimuli. So we pushed 6 different stimuli in a row, and for 10 seconds or so, we played these sequences of stimuli to the network and then asked whether any one rule would remember the sequences or how they would respond when we just played one item in that sequence to them. And we found that there's quite a variety of different responses. For example, this network here doesn't respond when we stimulate the background neurons, but does respond by predicting the next item in the sequence when we stimulate the first item. Here is a network that predicts all of the memories. In fact, it also has a response to the rest of the network, so we're not quite clear. It's a little bit dirty. Here's a network that has triggered replay. So if you play one sequence, the network will readily play the rest of this song for you, if you want to call it that. Although careful with the word song, I guess. And then here's another network that plays the sequence spontaneously. So you train it on a specific network, and it just keeps playing it for quite a while. In fact, some networks play this in the temporal scheme of the stimulus itself, and some accelerate it and play it quite quickly. What all the networks have in common, that very quickly after the training period, these sequence memories fade away completely. So after 5 minutes, you get sort of a slowed down version of the sequence, and then after 5 hours, there's really nothing left. We thought that was a cool thing because it meant that the networks weren't blowing up, but other people seem to think that's sad. In any case, you could think about building a second network that picks up representations of these sequences. So, just to finish this part, maybe I'll show you the learning rules that have a specific behavior. So in case of this set, these are all the learning rules that show sequential replay. These are all the learning rules that show non-sequential replay, and you have the other memory tasks here too: on responses, familiarity detection, novelty detection. You can start, if you squint your eyes, you can start to see trends here, or you can at least start to see interesting things. Why does non-sequential replay require STDP-like rules, but sequential replay has some sort of inverted Hebbian here? Why is there a STDP rule in the inhibitory to excitatory rule for sequential replay, and so on? So you can spend a long time looking at this picture and sort of going off into la la land. Of course, you can also do this differently. You can ask for a specific shape of a rule and say what other rules would fit with this rule. So in this case, we constrained the excitatory rule to look like a classical STDP rule that was able to perform novelty detection, and this is what we got. Again, for some people, it may be interesting to investigate these shapes. Something that can be said is that the more specific a task is, or the more constrained you are, the more rare the rules become. In fact, once we decided on some task, we had to rerun the SBI search algorithms to find more rules, because there was really only 1 or 2 of these rules around that were performing a particular task. I already told you, the novelty task, almost all of the rules do it. For the replay, it's really 1% of the rules that we found that are able to perform this. Most rules only are able to do 1 or 2 tasks. There's very few that do 3. There's none that do 4 or 5 tasks at the same time. So we don't have an all-American athlete, so to speak, that could do all of the memory tasks that we're asking it to do. And then there's also something to be said, of course, about how ridiculously simple our plasticity rules are. 6 parameters. There's no interaction between them other than their coactivity. There's no additional supervision. And so we thought maybe, we can use these unsupervised local learning rules as a basis for more sentient behavior. And when I say sentient, of course, I mean Pong. So we had a supervised learning algorithm that trained a readout neuron to move a paddle. So what you see here is the position of a Pong ball that is observed by a static network on the left and by a plastic network on the right, and you can already see the difference. In this case, you see this inhibitory shadow. Every rule set really behaves differently, but what they all have in common, and we haven't actually played with so many, is that they outperform the static network by a long shot when we're training a supervised learning algorithm to play Pong with the information from these networks about the position of the ball, largely because in the static network you can't predict the trajectory at all, but when we go to the plastic network we see that the learning algorithm can take into consideration not just one position but 2 positions, because there is a footprint of that ball, not just at its current location but also at previous locations, or sometimes at locations ahead of the ball. Okay, so here's the performance just in numbers. The replay rule that I picked out, or that Basile and Maciek picked out, I had nothing to do with it really, other than saying, hey, can you play Pong with this? Was able to perform at around a 70% level accuracy in hitting the ball, whereas the static network really couldn't do more than 3 out of 10. That is not to say that all rules are equally good. There are some rules that are actually worse than the static network in that they're making the network actively not be able to hit the paddle. Okay, what have I told you so far? It's possible to parameterize synaptic plasticity to search for good rules, good synaptic plasticity rules, that is. While good rules are few and far between, we could find thousands of rules thanks to SBI. These coactive rule quartets were selected for their ability to maintain stable networks, but they can perform a variety of memory functions that seem to take advantage of particular features of each learning rule. The discovered rules act only locally and may form the basis of or a substrate that more sophisticated supervised learning mechanisms can harvest their functionality from, as shown in the game of Pong. But what does this fact, that homeostasis is such a rich constraint, tell us about neurons? Why should neurons want to fire at a certain rate? Why would they want to have their environment stable? Why not below or above? And that's what I'm going to talk about for the second part. And there we're diving far away from neural populations, and we're going to focus on a single cell, really, and then later come back to the network. So, why neurons want to fire at a certain rate, in our opinion, in my opinion, and Chaitanya Chintalure's opinion, has to do with how neurons produce ATP. And I'll give you a very quick crash course on how neurons produce ATP. They produce ATP in their mitochondria. You didn't think you were going to do biochemistry today, but here you are. The ATP is produced from ADP at the ADP synthase. And it does so with the help of lactose and glucose that is being converted into pyruvate and then shuttled into the mitochondria. And here already I would like to make you aware of an idiosyncrasy that neurons experiences, which is that they cannot undo the pyruvate transition from lactose or glucose that other cells can. So all the pyruvate that they receive has to end up in the Krebs cycle, which churns and churns and churns and makes NADH at the end of it. And that NADH is being used to drive what's called the electron transport chain, to drive protons across the inner membrane of the mitochondria. And these protons are in turn used finally to drive the ATP synthase, without which the ATP cannot be milled. So this is your biochemistry in a nutshell. And I'd just like to iterate the points that are important here. All pyruvate must be used by the Krebs cycle. It cannot be converted into anything else. So neurons are running a very direct and very lean metabolism. The Krebs cycle is, by first approximation, always running as a result of that. Also because neurons can't fully control their metabolism. So lactose is being fed to neurons by their ambient astrocytes. So they're being force-fed in a way, and they have to make do with what they get. Depending on circumstance, So the electron transport chain will push protons across the inner membrane as much as it can, but sometimes it can't. So depending on circumstance, the electron transport chain also produces reactive oxygen species, and reactive oxygen species is quite bad for you, generally speaking. The limiting factor in the availability of ADP, limiting factor of this entire thing is the availability of ADP at the ATP synthase, okay? So the entire mechanism is being throttled at the very end of it. When ADP is lacking, the ETC begins to stall, but not the Krebs cycle, okay? So the Krebs cycle is running, you have one foot on the brake and the other one is on the gas. Okay, and what happens then is that these protons here are starting to accumulate on the outside of the inner membrane because the ADP synthase doesn't have any more ADP to make ATP out of. Okay? So what that means is that neurons can find themselves in two situations. They can find themselves in a situation where the electron transport chain produces more ROS than is healthy because a lot of ATP is being produced. But it's also possible that they find themselves in a situation where they experience more than healthy ROS levels because too little ATP is being used. Okay? They find themselves in a very classical non-monotonic relationship with ROS, and that makes for an interesting homeostatic challenge in this respiratory state space where on the y-axis you have the delta psi, so the gradient of protons outside, between inside and outside of the mitochondria, and on the y-axis, on the x-axis, the mitochondrial ATP availability. So when there is a lot of ATP around and the delta psi is high, so there's a lot of protons, you will get high ROS levels. But similarly, when there's no ATP around and also no protons around, there will also be a lot of ROS because the electron transport chain keeps producing and keeps trying to refill your ATP levels. Okay? So what can a neuron do to live in Ross Valley? Or to anthropomorphize, what would you do? This is not a situation unlike what some of you are facing when they live in New York City. I certainly faced that situation in my apartment in New York City, in that in the winter, the heat is always on. There is no way to turn down the heat. And that is similar to a slow ROS poisoning for a neuron. And humans in New York have developed quite a healthy habit to this sort of slow death by being cooked, which is to open the window, right? They open the window in the middle of winter, or some, like me, already even had to turn on their air conditioning to keep the apartment cold. So in other words, when there is too much ROS around, you should do one of two things, right? When there's too much ATP around, you should waste that ATP, and when there's too little ATP around, you should try to step on the brake and not use as much ATP anymore. And in fact, the latter part has been shown quite readily by multiple labs, so that you have ATP-sensitive potassium channels that will throttle your neural activity when neurons are running low on ATP. Okay, so putting it all together, if a neuron consumes too much or too little ATP, it will potentially poison itself with reactive oxygen. When a neuron experiences sudden or periodic loss of inputs, like you do when you close your eyes or when someone cuts out your eyes or what have you, the ATP consumption will decrease dramatically, not only through loss of spikes, but also through the loss of inputs. And it's estimated that your metabolic budget for neural activity is up to 70% of a neuron. Okay? In the absence of any regulatory mechanisms for the Krebs cycle, so that Krebs cycle running and producing NADH ongoingly, the neuron is in the situation that it must dissipate the energy. And other cells have a similar problem. Muscle cells have that problem, for example. Muscle cells solve that problem by converting ATP directly into heat. Neurons can't do that because neurons will cook themselves, right? If you produce heat inside of your cage, that's a bad idea. And you don't have a window like we do in New York City. But what they can do is periodically get rid of ATP in another way, which is to depolarize the membrane potential and spike. And so that's what we proposed. So if you want to conserve energy, put on a sweater, or another way is throttle your ATP output by way of a potassium channel. There's lots of evidence for that. Or you can waste energy by increasing ion channel conduction of, for example, a persistent sodium channel to depolarize, or in the extreme, to spike. And what you get from that is that you have some sort of a ambulant excursion of a neuron that sits somewhere high in ROS space and fires, and then goes down into an area where, by the consumption of ATP, the ROS levels are being relieved. Okay? And there's actually many, many ion channels that do monitor your oxidative state of the cytosol in neurons. So this is not a totally crazy idea. In fact, we have a long list of ion channels for you in the supplementary materials if you're interested. But what I'm just going to show you now is a proof of principle that this works in metabolically regulated networks. So we built a network quite like the ones that you've seen already, with the difference that in these linear integrate and fire neurons, we add an additional current, which was a metabolic current that monitored how much the neuron was spending on its inputs and how much it was spending on its spikes. Okay? And so when it was spending too little, the metabolic stress became quite high. When it was spending too much, similarly, but it would throttle output. So we emulated what I just told you, sort of a homeostatic response to metabolic state. And when we did that, we found that these networks, which initially, in this yellow part here, were driven by an outside current, very much like the networks I've shown you already, similarly to pretty much every other linear integrate and fire network that you see out there. In order to keep them alive, you have to push current into them, or spikes, or whatever have you. If you've ever worked with spikes, you know that this is a pain in the butt. But then when we turned that extra additional stimulus off, the network, like pretty much every other network, would stop firing, but the metabolic stress would then start to accumulate because these neurons were experiencing a situation in which there was too much ATP around, and all of the NADH that was being produced, supposedly, by this emulated mechanism, was being converted into reactive oxygen, right? And in that situation, the best thing to do would be to depolarize and fire off a spike to get rid of that ROS. And of course, if you fire a spike in a network that is depolarizing as a whole, as soon as the first spike is fired, all of its closest neighbors are also going to fire. And in fact, you see these avalanche-like starting points where in green you see metabolic spikes, and in purple you see regular network spikes that are simply driven by their neighbors. So the network starts to actually chug along again and finds eventually a steady state that I've now nicely hidden behind this panel, in which you find similar asynchronous irregular activity as before, but from an entirely internally driven mechanism. Okay? So this is quite nice, for us at least, because it reproduces a bunch of experimental results. It produces avalanche dynamics, it produces some neural dynamics that we see in the fruit fly, in which you can monitor ROS. There's many other knock-on effects that I won't tell you about that I would urge you to look at in the paper. It's in PNAS from 2 years ago, or about a year and a half ago. But it leaves us puzzled a little bit. At least it leaves me puzzled. And maybe it leaves you puzzled too, because I did the first half of the talk talking about spiking plasticity rules that produced homeostatic network environments. And then I told you in the second half that you don't actually need plasticity rules to do that for you, because metabolism is already going to take care of it, right? So we looked at this from a very neural-centric view, but it doesn't necessarily mean that actually these things are competing. They, in fact, might be collaborating with each other, in that plasticity rules might not have to pick up quite as much slack, and as a result of that, they might have more flexibility in doing other things. Something else that it raises, and that made our reviewers very angry, in fact, I don't think I've ever had angrier reviews, is that it raises a question as to whether the spikes were invented to talk, right? This means that spikes were evolved to get rid of energy. They're a product of wasting energy. And only later someone was like, oh shit, we can talk with these things, right? And our reviewers went off on a tangent and another tangent and 2 other tangents to the point that we were flabbergasted, because they were like, what about the evolutionary aspects of this? And we're like, we're not really touching this, but it was rejected multiple times because of this kind of anger that really sort of I found surprising, because it's just an idea, you know? In any case, I thought looking back at Heb might actually talk about how these two things can interact with each other in terms of plasticity, because Hebb actually originally said when an axon of cell A is near enough to excite cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, such as A's efficiency as one of the cells firing B is increased. Okay, so the metabolic aspect actually was included from the get-go, and maybe that's what Donald, not that Donald, wanted to point us towards. Early on. And of course, this is just a progress report, so we'll continue to work on this, and I'll hopefully tell you more soon at a different venue. For now, here are recommendations for posters. I already mentioned Juliana's talk, poster, and these two other posters were somewhere in there too. Memory as a byproduct of stability by Basile, and synaptic modulation, outperforms synaptic modulation by Ivan, as well as inhibitory synaptic plasticity. Learning by Maciek, and then finally selecting for local amplification by my graduate student Nicoletta. So, oh, and there's a talk by Adrienne Fairhall's lab right after this that's also using SBI, so stay tuned for that. And with that, thank you very much for your time, and I'm happy to answer any questions.
Okay, we have time for a couple of questions. So,
yeah, hi Tim. Thanks for the inspiring novel idea in the second part. I'm wondering what are alternative hypotheses that could explain that, and what are specific predictions that you're making based on that mechanism?
I didn't understand the question, I'm sorry.
Sorry. My question was, what are alternative hypotheses that could explain spontaneous activity patterns?
There's no other alternative hypothesis.
How could you distinguish them with experiments? What are
experiments I can tell you about.
Different predictions.
I can also tell you about sentience if you want an agency, but I won't. Maybe tonight. There's a bunch of experimental predictions actually we put into the paper, and that we're working on with some collaborators to figure out how you could regulate activity by way of, for example, hypoxia or something like that. So there's ways to manipulate the system. It's not totally easy because neurons happen to be very much in tune with their oxidative levels, and they try to avoid getting out of that trough by all means possible. And so experiments are often failing to get neurons outside of their comfort levels. I don't know where you went, Rainer, but that was my answer.
On the first part, you talked about 4 plasticity rules between E and I neurons. But at Allen Institute, we have been discussing about 100 neuron types. So in the end, 10,000 plasticity rules. How soon is that paper?
So I actually used to start this talk with a slide like that we have 140 rules, we've discovered 2, and it took us 50 years, so we still have 10,000 years of research ahead of us for the other 138. I mean, if you give me the compute, we can do it. But for now, we've used up, we used 1 million CPU hours for 4 rules, so it's going to take a little while.
But don't you think that there would be more other emerging principles?
So actually, to be completely honest, I'm hoping that we're going to find that they're all going to collapse into one rule to rule them all. Yeah, that would be great. We haven't quite gotten there, but give me another year.
Yes, on the same line, thank you, wonderful talk. So one suggestion to add another wondering to the list of that you raises by connecting the 2 parts of your talk is the following. If you have this homeostatic rule, which is one rule to rule them all, which is the cell tries to adapt to something, whether it's metabolic or communication with others, why bother and formalize 40,000 rules that are very limited, each of them, and they never will be fixed once you have this general rule, which is homeostatic adaptation, and that's it.
So you're right? I've shown you sort of 2 strands of my lab. One is very machine learning heavy, and the other one is very theory heavy. In my defense, the 1 million CPU hours that we used only used up half a transatlantic flight in terms of its CO2 footprint. So, we didn't burn the planet all that much, at least not as much as you guys did by coming here. But you're right. But the why questions are easy to answer in hindsight, right? If you don't know, you don't know, right? Science is finding a black cat in a dark room, and it's not even a cat. So this was the best we could do.
Hi. I had a question that is sort of related to the others, which is, so one, I guess there are a lot of reports that neurons don't actually need to rely on astrocytes for lactose, right? So they have an opportunity to at least, do more than just open the window, like you're saying. And then secondly, in the fly, there's examples of neurons that seem to use slow accumulation of ROS as a computational mechanism. And I'm wondering if you've started exploring ways that you can sort of learn your homeostatic plasticity rule in order to adapt to a task, right? So like a way to sort of generalize the phenomena that you're describing as a more adaptive learning process itself.
So to the second question, the fly stuff actually is what got us started. In fact, we started collaborating with a fly lab that asked us to build a model for this ROS accumulation, and then we realized, we never made it onto the paper, but we realized that we had something cool in our hands, and we started playing with it. As to that metabolic set point that the neuron should live at, I think we think that depends on the average metabolic output that the neuron has. So a neuron that fires a lot and uses up a lot of ATP wouldn't be much further out in terms of where the trough sits than a neuron that barely ever spikes, okay? And that is probably part of mitochondrial plasticity, actually. There's, maybe not recent, but work in the last decade or so that shows that mitochondria themselves can adjust the ionic flux of protons and so on across the inner membrane to actually adjust the overall efficiency of their function. But we haven't gone there very much yet.
Okay, we have time for one more question while the next speaker sits up.
Thank you very much. Do you think that this optimal ROS valley, does it depend on the neuronal type?
Yes, I think so. I think it depends on neuronal type, on size, on the where it sits in the signal chain. So I would assume that this is different for every neuron, even individually.
Thank you.
All right, let's thank Tim again. And our next contributor talk is from Chloe Lemont, who will tell us about the role of short-term plasticity in flexible behavior.
Is this good? Okay, good morning, everyone. My name is Chloe Lemont. I'm a graduate student in behavioral neuroscience in the Jackman lab. And this is my first Cosyne, so I'm really excited to share my work with everybody. Thank you. So I'll start out with a quick story. So the most important and most beloved piece of equipment in our lab is, of course, the mini fridge. It's where we keep all of our energy drinks, and I use it pretty much every day. And ever since I joined the lab, it's always been located at the end of the hallway. And so recently something awful happened, which was that we had to move it. And it's awful because now every morning, almost without fail, I will walk to the end of the hallway where it used to be and then get angry when there's just an empty spot there. And I think we've probably all experienced something like this before. Learning a simple association like where to find the fridge feels really easy, and yet unlearning a behavior and switching over to a new one can be surprisingly difficult. And this can also be the case in situations where the environment is changing very quickly and decisions need to be made based on recent cues and feedback. That flexibility to change our responses as the environment changes is fundamental to cognition, but we still don't fully understand all of the neural mechanisms that enable it. Many theories converge on the idea of rapid reversible changes to the response properties of neurons. And a key candidate mechanism for this rapid reconfiguration is short-term synaptic plasticity. On timescales of milliseconds to seconds, neurotransmitter release is modulated as a function of recent presynaptic activity. So some synapses exhibit depression, in which they're strong at baseline but become weaker with successive spikes, whereas others facilitate or strengthen with repeated activity but remain very weak if the synapse is quiescent. These two forms of short-term plasticity not only act as frequency filters for selectively transmitting different firing rates, but they also provide a temporary trace or memory of recent presynaptic activity, since their effects can last for hundreds of milliseconds before returning to baseline. To illustrate how these two processes could reconfigure the response properties of a circuit, we can imagine one input neuron connected to two output neurons. If each presynaptic terminal expresses a different short-term plasticity profile, then rapid presynaptic firing would temporarily boost one pathway while weakening the other, and the opposite would occur at lower firing rates. Now, these are two extreme cases, and in reality, the circuit's synaptic or hidden state could exist anywhere along a spectrum between them. So if we added an input to the presynaptic neuron, we see that the effect of its activity on the two different output pathways would depend on the synaptic state. And then we can get a different response to the same stimulus without any permanent changes to the circuit. And this motif, where a principal cell expresses short-term plasticity in a way that is specific to the identity of the postsynaptic target, is actually seen across the brain, which tells us that this is developmentally regulated and likely serves an important function in a lot of different brain regions. Although these ideas have been around for decades now, we don't have much direct experimental evidence from awake behaving animals because our methodology is still limited. The techniques that are currently available for observing moment-to-moment changes in synaptic strength in vivo are generally difficult and typically have low yields. However, our group has identified a key molecular mechanism of short-term plasticity that can be easily manipulated. So by sensing the residual calcium that builds up during repeated high-frequency firing, a protein called synaptotagmin 7, or SYT7, temporarily increases the release of synaptic vesicles, which leads to facilitation. And when we knock out SYT7, we abolish facilitation across the brain, but without affecting baseline synaptic function or triggering major compensatory changes to neurons, including the expression of long-term forms of plasticity. So this allows us to directly probe the role of facilitation in behavior and in neural function. Today, I'm going to cover our preliminary work using recurrent neural network models to link behavioral and neural phenotypes seen in SYT7 global knockout mice. So the first thing we examined is how losing facilitation would affect behavior. And because SYT7 is so widely expressed across the brain, we initially expected big changes, so we're surprised to find that knockout mice are actually pretty indistinguishable from their wild-type litter mates in that they're healthy and they breed well, and they don't show any gross motor or anxiety level phenotypes. So we're able to train them to perform operant tasks for water rewards. And because short-term plasticity has been suggested as a way for neurons to maintain information across brief windows of time, like in working or short-term memory, we started out by testing a lot of different delayed matching tasks, like this one, for example, in which an animal has to remember a previously cued reward location across a variable delay period. And we find that wild-type and knockout mice actually perform equally well in this task across a range of different delay lengths. The data here is showing performance at a 0-second delay, but they're good up to 30 seconds of delay, suggesting that facilitation is not actually required for learning to associate actions with rewards or to perform this type of short-term memory-guided motor planning. Another version of the task is a non-matched sample, where all of the task stimuli are the same, but the rewarded action is inverted between the trial types. So a left sample would mean the right port is rewarded. And knockout mice can learn this task as well, even when we increase the sample locations to 5 different ports. However, if we train mice on match to position, and then change the task rule from match to non-match and continue to train the same mice on the new rule, we find that knockout mice take twice as long as wild types to reestablish criterion level performance on this reverse task. So after seeing this really large reversal learning impairment, we also wanted to know whether it would generalize to a more rapidly changing environment. So we also tested a task with repeated within session reversals, where the rewarded and punished port would swap anytime the mouse made at least 8 correct choices in 10 consecutive trials. So there could be multiple reversals per session. And we see impaired performance and knockouts in this task as well. So we're seeing that a knockout of SYT7 impairs reversal learning on both these longer timescales and these short timescales. Altogether, suggesting that while the basic ability to learn has remained intact, the flexibility to update or discard established associations seems to be impaired when synaptic facilitation has been globally disrupted. So to explore how facilitation might support this type of flexibility, we turned to recurrent neural network modeling. We used modified vanilla networks with both excitatory and inhibitory neurons, and the key modification was that recurrent weights were modulated by short-term plasticity during a trial, using a rate-based implementation of the well-established phenomenological model of activity-dependent facilitation and depression by Sodix and Markram. We made two types of networks, so wild-type networks with a 50-50 split of depression and facilitation at hidden layer connections, and knockout-type networks exhibiting only depression, so more similar to what a SIT7 knockout brain would be like. Both types of networks were trained on a match-to-position task with one hot encoded stimuli to mimic the behavioral task that we trained mice on, and backpropagation through time was used to minimize the difference between the network's output and the target output. And since baseline weights between recurrently connected neurons were modified during learning but then fixed, while short-term plasticity dynamically modulates those baseline weights during each trial, this lets us model the long-term synaptic changes that would happen during learning while also capturing the contribution of short-term plasticity on the timescale of single trials. The two types of networks learn this task at similar rates and achieved similar levels of accuracy. However, once we fixed their recurrent connections and tried to retrain only the input and output weights on a reversed rule, so pre-training on match, fix the hidden weights, and then train on non-match, we find that the wild-type networks adapted successfully, but the knockout networks could not reach the same level of performance on the reversed version of the task, which is mirroring the behavioral data that we have from mice. So since the recurrent layer weights remained fixed while learning the second task, we interpret this that the wild-type networks were better able to remap the connections to and from their hidden layer to apply that learned hidden layer structure from the pre-training on match to the new non-match rule. We also examined how sample identity was encoded in each network's hidden layer during the delay period of the match to sample task, on which both models performed with equal accuracy. Looking at dimensionality-reduced principal component space plots, we can see the activity of all hidden layer neurons, which here have been combined into pseudopopulations for all 10 replicates of each network type. And we find that the hidden layer activity of wild-type networks actually shows very high sample separability, whereas knockout networks show much less distinct representations in the hidden layer. And this finding was surprising because we actually expected that having multiple types of short-term plasticity might make wild-type networks even better at activity-silent storage, for example, than knockouts. But instead, it seems that the lack of facilitation makes the delay period even quieter, with more muddling between the samples as a result. So having replicated our behavioral phenotype in these models, we lastly wanted to test the model's predictions by recording neural activity in behaving mice. So after surveying multiple brain regions using local field potential recordings, we eventually chose to focus on prefrontal cortex because we know that SIT7 is highly expressed there and also been demonstrated in vitro to drive facilitation in this region. Looking at putative pyramidal neurons, we didn't find any baseline changes to average firing rates or to the amount of burst firing. And we already know from in vitro studies that knocking out SIT7 doesn't affect intrinsic excitability. So this in vivo data suggests that we also aren't getting a huge network-level compensatory effect either. When we standardize activity across the recorded population of neurons and then look at average trends while mice are learning to associate lit ports with rewards, we see that wild-type PFC generally shows distinct population-level activity associated with rewarded versus unrewarded pokes, while knockout PFC shows very little differentiation between these two outcomes, despite both genotypes acquiring the reward association at a similar rate. So we extended this comparison to look at all the subphases within match-to-position task trials as both genotypes learn this slightly more complicated task. So combining all neurons from each genotype into a pseudopopulation and then again visualizing time-averaged activity, this time around each unique phase in principal component space, we see that similar to what was predicted by our models, the representation of task variables in wild types showed much higher separability in state space than knockouts did. And so you can see there's unique state spaces for some of the different task phases, and then we saw additional separation for left and right trials in the punish phase, for example. Meanwhile, in knockouts, these differences were very much diminished. So these findings were consistent with our model's prediction that facilitation might increase the distinctness of different task-specific neural activity states. So to bring it all together in the context of our driving question, our data suggests that while basic learning doesn't require SIT7-driven facilitation, flexible updating might be impaired when it's been disrupted. We see that SIT7 animals show behavioral deficits in tasks requiring adaptation to new rules. Modeling shows that removing facilitation impairs the network's ability to remap its weights to solve a new task. And PFC recordings showed reduced separation between neural states corresponding to different task events during learning. And so in the future, we plan to expand this work to include conditional knockouts, explore other brain regions, and perform recordings during flexible behavior. And lastly, I'd like to thank my mentor, my lab mates, and our collaborators for their support. We're a very small synaptic physiology lab, so a lot of this work was only possible through collaboration. And I'd love to invite anyone interested in collaborating to please reach out, and thank you all for listening.
We have time for a few questions.
Hi, it was a really beautiful talk. Thanks a lot.
Thank you.
So I was curious, you might have showed this and I missed it, but you showed that when you train the RNNs on like the second version of the task, the ones with STD and STF like learn it much faster than the ones with just STD.
The same number of training epochs, they can all reach criterion, but accuracy in terms of how much more they're outputting the correct sample than the incorrect sample, they have higher accuracy in networks with both facilitation and depression versus the knockouts, much lower accuracy.
Cool, okay. I was wondering where networks with no short-term plasticity fell in that performance ranking.
Oh, so like no short-term plasticity, all just fixed?
Yeah.
So they tend to, without the short-term plasticity, the networks are much simpler, and I actually find they're kind of just easier to train in general, and will learn tasks pretty much fine.
Yeah. So in your like restricted framework, the ideal thing would be to actually have no short-term plasticity, but then if you have to have it, it's better to have both than to just have the depression.
Yeah, because we're trying to model, so in a SIT7 knockout mouse, for example, like there will still be depression everywhere, and you know, synapses that depress but express SIT7 would actually show even more depression. So like synapses that have frequency invariant firing when we knock out SIT7 would then just kind of drop down to the super uniform response. So we felt that the knockout type network was a better comparison to a network with and without, because the dynamics just look completely different with a fixed network.
Got it, cool. Thanks a lot.
Thank you so much.
All right, let's thank Chloe again. And our next speaker is David Bell, who will tell us about discovering plasticity rules that organize and maintain neural circuits.
Okay, hello everyone. Thank you to the organizers for the opportunity to speak. I'm really excited to be here at Cosyne and to present some of my recent work. And I also now have the distinct pleasure of following two really excellent talks on meta-learning plasticity rules and self-organizing circuits via plasticity. So good luck to me. Okay, so an idea that's heavily influenced me lately is that we can think of the genome as encoding a prior on learning through mechanisms like large-scale structure, cell types, and plasticity rules. These mechanisms likely interact to produce brains that are primed to learn rapidly and robustly. And today, as is the name of the session, I'm just going to focus on plasticity rules. Okay, so here's a hypothesis for how the genome might encode priors via plasticity. Perhaps plasticity rules can self-organize key computational motifs before an organism has substantial experience. So for example, when a zebrafinch is learning its courtship song, this process is famously scaffolded by a timing sequence in area HVC that is believed to self-organize during development. In similar fashion, Drosophila navigating the world might be aided by intrinsic plasticity that allows it to organize some sort of continuous attractor network, allowing it to keep track of its head direction. And mouse circuitry governing decision-making while foraging might begin with intrinsic plasticity that sets up some kind of saddle dynamics. So circuits that are self-organized without substantial experience in the world and maintain themselves in the presence of noise can serve as a powerful scaffolding for subsequent learning. And the question I'm going to focus on today is, can we and how might we find plasticity rules that self-organize and maintain such motifs? Today I'm going to focus on the very simplest case, timing representations like sequential dynamics as a proof of principle, and I'm going to ground this in the example of the zebrafinch. As many already know, the zebrafinch learns a highly stereotyped courtship song that is seconds to tens of seconds long. There are two nuclei known to be essential to song production. These are area HVC and RA. Pioneering electrophysiology work revealed that the RA projecting excitatory cells within HVC fire in a sparse, song-locked fashion, while an interneuron population fires tonically during song. These dynamics are thought to provide some sort of timing scaffold for song. So here's the putative connectivity of the circuit, very, very simplified. The RA projectors essentially wire up in some kind of feed-forward excitatory sequence, while the interneuron population implements global inhibition. HVC is known to be remarkably resilient. It's known that song can recover following perturbations to HVC, such as transection, viral silencing of constituent neurons, and partial ablation of inputs, which is quite remarkable. So the slightly narrower question I'm going to focus on today is, can we find plasticity rules that self-organize these dynamics and are robust to perturbation? To do that, I'm going to adopt a method that Tim Vogels and his lab has been pioneering that actually began with Yoshua Benjo. So I guess I found that 2020 paper about kind of works pretty inspiring. The additional idea from this work came from that paper. And the idea is to parameterize a synaptic update rule with a set of basis functions, these f sub k, which we will take to depend on things like the pre- and post-synaptic activity of the synapse, as well as its size. These basis functions are weighted by a set of coefficients and additionally have tunable time constants. We can then take a parameterized learning rule and simulate its effect within a network of firing rate neurons, evaluate the resulting dynamics with respect to some loss function, and then use that loss with a supervised learning procedure to adjust the coefficients and time constants. And we'll do this iteratively until the loss converges. To ensure we learn rules that are robust to biological noise, we can simulate networks with random synaptic turnover, a process by which existing synapses die and new ones are born with some small probability. So how do we use this process to learn timing representations? In the simulation phase of meta-learning, we provide a subset of the neurons in the network with a strong kick of excitation at time equals 0. The network will respond with some dynamics, and then we can grab the firing rates at random moments in time and attempt to decode how much time has elapsed since the stimulus was presented. In reality, we will stimulate the network many times, and then we'll use a late set of activations to train the decoder and an even later one to test it. And this decoder error becomes the primary term in the loss function. To this, we add penalties for activity and synaptic change, which we think of as energetic constraints on the plasticity being learned. And I'll mention that for the time being, we'll constrain ourselves to learning a rule just on the excitatory to excitatory synapses within this network. Okay, so once meta-learning has converged, what we have is a synaptic update rule that can take a network with initially random connectivity and disorganized dynamics, and can shape it into a network that produces stereotyped sequential dynamics like we see in HVC. And when we sort neurons by their typical firing time, what we find in the connectivity matrix is that a sparse feed-forward structure emerges, as HVC is thought to be structured. So the learning rule in general will have many of these coefficients, these CK, be non-zero, but we can refit these coefficients in order of their importance in order to attempt to extract a minimal learning rule. And what we extract in this case is a temporally asymmetric generalization of Oya's rule. So here's Oya's rule, where xi is the presynaptic activity, xj the post, and wij is the synapse size. And here is the minimal rule we distilled from meta-learning. These C here are positive coefficients, and the tildes denote convolutions with decaying exponentials. So xi tilde, for instance, is a smooth filtration of the presynaptic activity. The first two terms here implement a form of asymmetric, temporally asymmetric Hebbian learning, while the latter term prevents synapses from getting too large in an activity-dependent fashion. Crucially, this rule does not impose any hard constraints on connectivity, which will be important in a bit. So it's already known that there are plasticity rules that can self-organize sequential activity. There's a rich literature on that. So we wanted to focus on the resilience aspect of this rule. We tested the learned rule's ability to maintain a timing representation when we introduced synaptic turnover to the circuit. We compared our rule, our meta-learned rule, to another from the literature which imposes hard bounds on the size of individual synapses, as well as a constraint on the total amount of connectivity onto individual neurons. We found that the performance of the two rules was similar in the absence of synaptic turnover, but that the meta-learned rule excelled when the network structure continually shifted. We attributed this to the absence of hard bounds on the connectivity, which allowed the meta-learned rule to flexibly rescale synapses as needed. So we can also use meta-learning to explore the roles of multiple interacting plasticity rules learned on different sets of synapses, as Tim just showed. And in HVC, recent work from the Lois Lab and others has revealed that the connectivity from the excitatory population to the inhibitory and back can also evolve in response to disruptions. So therefore, we learned synaptic plasticity rules on all three sets of synapses here, E to E, E to I, and I to E. It's known that interneurons in HVC do not heavily interconnect. We found these rules, again, could organize feed-forward structures, and also discovered that this additional plasticity better maintained timing representations through synaptic turnover. And we were curious as to why, and so our hypothesis was that this additional plasticity would allow individual excitatory cells in the sequence to better maintain their typical firing pattern and timing through synaptic perturbations. So to illustrate this, imagine we have a sequence. We can focus on just a single neuron to begin with. It will get some input from the chain, and will have some typical response as a function of that input. When synapses turn over, this will affect the effective input that the neuron sees and therefore shift its firing pattern and timing. And so ideally what would happen under plasticity is that this neuron would be able to compensate for that altered input by regaining its original firing pattern and timing. Okay, so to simplify things for ourselves, we can characterize a particular neuron's response to this input via three variables: the duration, peak amplitude, and delay. If we plot this response in the space of two of these variables, the amplitude and duration, it might look like this. We can now imagine that many different synaptic perturbations resulting from synaptic turnover result in different effective inputs that a neuron sees, and we can plot all of these responses in the space I just showed you. We now ask what happens when, in a simplified model, only the excitatory inputs are allowed to evolve under some kind of E to E plasticity rule. So this one's homeostatic. So here are the initial responses, and after plasticity shapes the excitatory inputs, what we see is that all these responses will evolve to essentially a stable line shown here with the blue stars, indicating. So ideally, all these responses would have all converged to one point in space, indicating complete compensation for the altered input, but this does not happen under just an E to E plasticity rule. If we look in the space of amplitude and delay, the picture is similar. So now, what happens when we consider two plasticity rules in this case? What we find instead is that two forms of plasticity are better to better constrain the amplitude and duration of the response. So all these initial responses will almost evolve to a single point. This is similar to what Tim Vogels showed in 2011. But an additional surprising feature is that the timing of the response is also stabilized, indicating that this interneuron plasticity can better maintain both the firing pattern and timing of neurons in the network. Okay, so to conclude, we use a meta-learning procedure to find a temporally asymmetric generalization of Oya's rule that self-organizes timing representations. This rule is robust to synaptic turnover because it does not impose hard bounds on connectivity, allowing synapses to rescale in response to changes in activity. Learning distinct rules for these different sets of synapses leads to even more robust representations of time, and we think this occurs because we showed that in a simple model, additional forms of plasticity could support better timing representations by allowing individual neurons to maintain their firing patterns and timing. I think on whole, we demonstrated the viability of the meta-learning framework as a way of learning plasticity that self-organizes and maintains circuits. We're now very interested in finding plasticity rules that can self-organize and maintain continuous attractors, particularly when the connectivity is not symmetric. All right, so with that, I'd like to thank my lab and advisor, Adrienne Fairhall, collaborator, Alison Duffy, as well as colleagues at the Lois Lab who really inspired this project, and finally the Brain Initiative and Simon's collaboration for the Global Brain for funding this work. Thank you.
Okay, we have time for 1 or 2 questions. I think Juliana has one.
Hi. Running fast. Okay, so these are rate-based rules, right? But
yeah.
in HVC and in the birdsong systems, the neurons are spiking, so they generate these precise sequences of spikes. So I was wondering if you're already working on thinking about this, how this could generalize to sequences that are produced for spiking neurons, potentially with spike timing dependent plasticity rules.
It's a good question. It's one we got from reviewers. I think we knew wandering into this project that tackling spike-based rules was going to be a lot more complex, so this was sort of our first order take on this, and definitely something to, well, Tim is already exploring this.
Thanks.
Yes, 2 things. One, in response to that prior question, there's been other work that shows that similar heavy rules that are applied to rate base can be transferred to STDP, so I'm not too worried about that. The real issue is, of course, that the zebrafinch is the outlier in the vocal communicating birdsong species, in the fact that it is the only species that has a linear sequence of syllables. Most birds have a much more complicated and probabilistic sequential structure, and it's not at all clear how these types of learning rules would apply to those types of sequences. Could you comment on that a little bit?
I think that's very valid. I think in other songbirds, often there's a syllable structure, and then the intersyllabic transitions can be more probabilistic. So it's possible that different rules govern sort of within syllable structure versus intersyllable structure.
Great, thank you.
All right, let's thank David one more time. And our final speaker of the session is Catherine Wang, who's going to tell us about the emergence of robust persistent activity in the premotor cortex across learning.
Testing. Okay, last talk of the session. Hi everyone, my name is Catherine. Thank you to the organizers for this opportunity and for you all for being here. Today I want to talk to you about circuit mechanisms of persistent activity. Persistent neural activity underlies many key cognitive functions like short-term memory. During short-term memory, neurons in frontal cortex show persistent firing. This persistent firing allows the brain to temporarily maintain information to bridge past and future events. To get persistent activity, networks need to have 2 basic properties. First, individual neurons have short time constants. To sustain seconds-long activity, a key motto specifies that neurons need to form recurrent excitatory connections in a network. This recurrent amplification gives rise to persistent activity. However, just recurrent amplification is not enough, as networks are typically fragile to perturbation, whereas the brain is robust. This resilience to perturbations is best demonstrated in clinical examples like stroke or neurodegenerative disease, where the brain may lose a lot of neurons, but can still largely retain a normal level of cognitive function. So one way to get robustness is through gating. If recurrent circuits are compartmentalized into modules, then local disruptions would not affect the whole network. The question we want to ask is, how does the brain implement these 2 mechanisms for persistence and robustness? We study persistent activity in the mouse using a short-term memory task. So in this task, the mouse senses a cue with its whiskers, maintains a short-term memory during a delay period, and reports its choice with a directional lick. This short-term memory depends on a frontal cortical region called the anterior lateral motor cortex, which I will refer to as the ALM. In the ALM, neurons have persistent activity that is selective for lick direction. This persistent activity is also robust to perturbation, so when the ALM is optogenetically inhibited, the activity recovers. This robustness is mediated by a modular organization across the ALM hemispheres, where each hemisphere can independently maintain persistent activity and gate out perturbation in the other hemisphere. However, studies like this are typically done in highly trained animals, which leaves open the question, how do these robust persistent representations emerge? So earlier I introduced recurrent amplification and robustness through gating as 2 circuit mechanisms for robust persistent activity. The question we want to ask is, how do these circuit mechanisms arise? One possibility is that network features like gating and persistence are innate to the brain, and these provide the initial conditions for persistent representations to be learned. Alternatively, the brain could start from a blank slate, tabula rasa, and all connections are plastic. So what is the network initialization for learning robust persistent representations? To examine this question, I used 2-photon calcium imaging to study how ALM activity changes as mice learn the short-term memory task. In this setup, mice progress from a naive to learning to expert stages over weeks, measured by the proportion of correct trials. Longitudinal imaging allows me to consistently identify the same field of view and neurons throughout learning. So on the right here, in an example field of view, the matched neurons are shown in green across the 3 stages. Okay, let's look at what happens at the level of neural activity. So let's look at one example neuron overlearning. Here on top, I'm showing you the fluorescence traces of a neuron sorted by right and left trials, and the bottom shows the average traces as blue and red. This first neuron is quiet in the naive stage, and overlearning, selective persistent activity emerges during the delay epoch. For contrast, here is a second neuron that is trial-type selective in the response epoch. This selectivity is already present in the naive stage and is stable across learning. At the population level, we observe an overall increase in task-selective neurons, quantified here on the y-axis, but particularly during the delay period. So where do these selective neurons come from? These bars here summarize the data I just showed you. So in the naive stage, there are virtually no delay neurons, but already some response neurons. Over learning, the delay neurons are recruited from the non-selective pool, as are the response neurons. From learning to expert, delay neurons are mostly retained, but at the same time, some delay neurons drop off while others are recruited. So this turnover suggests continued refinement of the short-term memory representation. To look at the impact of these single neuron changes on population information coding, we turn to a high-dimensional approach. So consider an activity space where every dimension corresponds to one neuron. In this space, lick right and lick left trials are distinct trajectories. We can find the choice coding vector by maximally separating these traces at the end of the delay. If we project the trial-wise activity onto this choice vector, we get the following figure. Here, right and left trials are colored as blue and red, and the thick lines show their trial averages. So in this case, I estimated the choice vector in an expert animal, and here I'm looking at the activity projected on this dimension in the same expert animal. You can see there is good separation of the trial types through the delay period, meaning that this dimension carries persistent trial type information. Now, because I'm tracking the same neurons, I can look at the activity on the same dimension when the animal is in the learning and naive stage. What we see is that this dimension was not persistent in the beginning, and this emerges over learning. To quantify this effect over animals, we decode trial type from the choice vector separation at the end of the delay, and overall fields of view, the decoding accuracy improves. So at the same time, we noticed something else unusual over learning. In the learning stage, we saw variability in the activity of neurons. To illustrate this, I'm showing you the mean delay activity of a single neuron in the learning stage. So you can see here that it's sporadically active over the trials, but in the expert stage, however, this activity becomes more consistent. At the population level, we can plot the activity of neurons on the y-axis across trials on the x-axis. And you can see a vertical banding pattern across the neurons, meaning that these neurons are co-active, but not on every trial. To probe deeper into this variability, my collaborator Taiga Abe, a postdoc in Shaul Druckmann's lab, fit a topic model to the neural data. So topic models are well suited to discovering cluster structure within the data. In this case, it's able to find clusters across neurons and trials. In other words, we can look for neurons that covary in the trials that they covary on. So when Taiga applied this clustering method to this learning stage activity, he found 3 distinct clusters of neurons that were coactive on different trials. To help you visualize this better, I'm showing you the same data, but sorted the neurons in trials by their topic model cluster assignment. So you can see now that there are 3 clusters and neurons are active in the different clusters. And by contrast, in the expert stage, the activity becomes more consistent on every trial. These delayed neurons form the choice coding direction. The fact that we can find this clustering structure initially suggests these clusters gave rise to multiple inconsistent coding directions. Then they become a single consistent direction over learning. With this data, what we see is that in the naive stage, there are no persistent representations of choice. As mice learn, these persistent representations emerge and become refined. So overall, this suggests that recurrent amplification forms over learning. What about the robustness of this network? Is gating built in or also learned? To probe network robustness, we use an optogenetic approach to silence the right hemisphere while imaging from the left. So this is like lesioning a portion of the cells in a network and measuring the network response. Let's look at an example neuron. The left here shows a control trial activity. So when a transient perturbation is applied in the early delay, the trial type selectivity is abolished. This is the activity at the learning stage, but if we look at the same neuron in the expert stage, the activities become robust to such perturbation. Here is a summary data across all neurons. This is a trial type selectivity in control trials shown in gray. So the selectivity is initially weak in naive mice and emerges over learning. The red trace here shows the selectivity in perturbation trials. So in the learning session in the middle, the activity is not robust to the perturbation to the other hemisphere. So the selectivity is abolished by the perturbation and doesn't recover through the delay. And in the expert animal, the selectivity is retained. This emergence of robustness also has an impact on behavior. So we can quantify robustness as a fraction between selectivity in control and perturbation trials. Using this measure, we find a positive correlation in the increase in robustness with better behavioral recovery to perturbation. So in other words, in sessions where neurons were non-robust, behavior was also affected. So to summarize so far, we found that both recurrent amplification and gating are learned through experience. This suggests that the brain starts from a blank slate, first learns recurrent amplification, then robustness through gating. So how is this emergence of gating implemented? One possibility is that long-range connections are fixed. Local plasticity allows each hemisphere to learn gating. This scheme is commonly used in training RNNs, where long-range inputs are fixed and local weights are learned. Alternatively, long-range connections could also be plastic. This change is more powerful, but requires coordination between local and long-range learning. So what is happening in the brain? To measure coupling, we identify single neurons that are significantly impacted by the contralateral hemisphere perturbation. We quantify this activity change at the time of perturbation to measure coupling directly. So here's an example neuron. You can see that the activity level shown in red is significantly reduced by the perturbation. We can quantify the prevalence of these neurons, and across learning, there's about the same amount of neurons that are susceptible. This suggests to us that the coupling strength between hemispheres are constant, which is interesting given the emergence of gating over learning. So here we might think that long-range connections are fixed. But actually, that's not the case. Unexpectedly, we find neurons susceptible to contralateral perturbation are not the same over learning. So in this flow diagram, you can see that there is a lot of turnover of susceptible neurons across learning. This tells us that the coupling between brain regions is changing. And to examine the impact of this change, we look at network coupling in population activity space. So at the population level, we can look at activity states in control and perturbation trials. The change is the impact of the contralateral hemisphere on activity. We call this the input vector. So here, activity projected on this vector captures a contribution from the contralateral hemisphere, which is strongly impacted by the perturbation. We can calculate this vector in the learning and expert stage and ask if they are oriented the same way. To do this, we calculate the angle of these vectors over learning, and here we find that they are very different. So across fields of views, the dot product is generally far from 1. So this rotation of the input vector suggests a change in the communication subspace over learning. These data rule out the first hypotheses. Instead, we find that long-range connections are also plastic over learning. This means that brain areas are communicating along different dimensions over learning, and we also then know that persistent activity evolves in distributed brain areas. This discovery leaves us with some food for thought questions, like how do brain areas coordinate with each other when connections are changing both locally and across areas? And how does the communication subspace change, and how do these changes relate to local coding vectors? This is where I'll leave off the talk. I want to say a quick thank you to my amazing PI Nuo and our wonderful collaborators Shaul and Taiga, to our funding sources, and I'm happy to talk about this work during the conference and happy to answer questions now.
Right, we still have time for a few questions. Maybe while we wait, so in the last part that you showed the rotation of the input vector, how would you distinguish a change in the communication subspace from just the other hemisphere exploring a different dimension in the in a static communication subspace?
Yeah, so that's a really interesting question is like, what are the two hemispheres doing over learning? Because we're not able to, at this time, image from both hemispheres. So I guess one way to address that question is to look at like what exactly this input vector aligns with in terms of, we know we have the choice dimension, and one thing that we do find is that it seems like the input vector becomes more aligned to the choice dimension over learning. Also to address kind of what the other hemisphere is doing, I'm currently performing dual like neuropixel recordings in both hemispheres over learning to see how the internal like dynamics change.
Okay, well, if there are no other questions, let's thank Catherine again and the other speakers. And we have a lunch break now and the poster session, and I think the next session in this room starts at 4:15. I told this person to come back to sign.